Hello, everyone. My name is Gerard Gatch, and I'll be the moderator for today's webinar on the technology and economics for the purification of oligonucleotides by twin column continuous chromatography. This is one in a series of more than 10 webinars on multi-column chromatography for applications in the production of protein therapies. We understand that you have limited time and came here to hear the content, so we'll forego formal introductions of our company until the end of the webinar. Only a few housekeeping notes and we'll commence with the presentation. Today's presentation is approximately 30 minutes long, leaving plenty of time to answer any questions you may have. Okay, so our agenda. Today we'll be going over multi-column continuous chromatography and how it works, in particular MCSGP. Thomas will present a case study. We'll talk about the economics of applying MCSGP. And then we'll have a summary and open it up for questions. Today's presenter, Thomas Müller-Spaeth, PhD, holds the position of Chief Technology Officer at YMC Chromacon in Zurich, Switzerland. Thomas is the inventor of more than 10 patents and has authored and co-authored more than 30 scientific articles and book chapters on continuous chromatography for biopharmaceuticals. Thomas frequently presents at international conferences as speaker and also co-chairs workshops on continuous chromatography. Please welcome Thomas Müller-Spaeth. Thomas?
Yeah, so can you still hear me? I guess so, I hope so.
Very
Otherwise,
well.
Please let me know. Haha, nice joke. Okay, so so thanks for the introduction, Gerard, and I'd like to also welcome you. And today's talk will be about MCHCP. So MCHCP is a technology that uses twin columns, so two columns of the same type. And today we will focus on its application to oligonucleotide purification. So firstly, what is MCSGP? It's first of all an awesome acronym, right? It can be used as a password for your computer. We keep saying that. Some people actually do. Their accounts never get hacked. MCSGP stands for multi-column countercurrent solvent gradient purification. So two columns, it is a countercurrent process. It can do linear gradients as opposed to other continuous chromatography techniques like simulated moving bed chromatography, 4-zone SMB that you may know from the past from applications of chiral to chiral molecules. Okay, so the challenge that MCSGP is addressing is mainly the removal of product-related impurities, which actually elute very closely with a main compound peak that is eluting in the center of the chromatogram. So we have weakly adsorbing and strongly adsorbing impurities in the front and in the back of the main peak, which we want to separate. And now in single column chromatography, we are facing the challenge that we have the overlapping portions in the front and in the back that contain a large amount of product. And if we want to obtain product with high yield, we actually run the risk that we are including these impurities in the product pool. So we either have a very pure product from a center cut in a very narrow fraction that corresponds to high purity but a low yield, or we have a wider product fraction including the impurities in the front and in the back, so we get a high yield but a low purity. So this is a trade-off between yield and purity that we are facing in single column batch chromatography. Now MCHCP can actually help us to alleviate this problem. And what it does is it internally recycles impure side fractions and recovers the product from the impure side fractions. And like this, it can obtain high yield and high purity simultaneously. We have to say also that the trade-off that in single column chromatography between yield and purity becomes worse when you try to increase the load or when you try to increase the flow rate. Both will lead to larger overlaps, first increasing load because just the peaks become broader through the increased load, and through the flow rate we're also increasing dispersion and diffusion limitation so that the peaks will become broader. So the situation with yield purity trade-off in single column chromatography actually becomes worse when you try to improve the productivity. Now, again, in MCSGP, we can move to the upper right corner and get high purity and high yield simultaneously. So in single column chromatography, we have to decide what to do with these impure side fractions. So in many biologics applications, the side fractions are discarded. In oligo and peptide purifications, these side fractions can be, let's say, subject to rechromatography to some extent. So they are reprocessed, and so in an attempt to recover the product in the side fractions, but this leads to a reduction of overall productivity, and also it means that the side fractions have to be analyzed and stored, which represents a significant effort, and especially with sensitive side fractions, it may not be feasible. So in continuous chromatography in MCSGP, we internally recycle the impure side fractions automatically to get a higher yield for the product. So we can do center cut purifications, we can run linear gradient. That's a counter-current process, which means it has improved separation power, and it does the automated recycling of the impure side fractions. So let's see how this process works more in detail. So again, we have our purification challenge here, represented by these three peaks that are coming off, eluding in the chromatogram. So we can separate this chromatogram here in 5 different zones based on the presence of pure impurities, an overlap between the impurities and the product in the front, and the pure product, an overlap between the product and the strongly absorbing impurities in the back, and the strongly absorbing impurities eluding at the very end. So this mixture now is loaded onto the first column. So these 2 columns are actually contain resin material of the same type, and could be, for example, an ion exchange material. So we load this mixture now onto the first column, and we start to run a linear gradient. We elute the weakly absorbing impurities using this linear gradient. So this is essentially the same like in single column chromatography, but then comes the overlapping portion between the product and the impurities here. And before this actually elutes from the single column, we interconnect the 2 columns to transfer this portion of the chromatogram from the first column into the second column. So and then we inline dilute this stream on its way to the second column, so that this overlapping portion, the compounds in this overlapping portion, get reabsorbed at the entrance of the second column. Okay, so this is the side fraction that we keep inside the process. So now at the outlet of the first column, we only have the strongly absorbing, we have the product coming off next. So we resolve the interconnection between the 2 columns, and we continue the linear gradient on the first column, and we elute the product from the first column and collect the product from the first column. At the same time, on the second column, we load new starting material onto the column, which then stays at the column entrance. Now, looking again at the first column, the next portion that would elute from that first column is the overlapping portion of the product and the impurities, the strongly absorbing impurities. We want to keep that fraction inside the first column, inside the process. So we elute it from the first column. So we run a linear gradient on the first column. And again, we inline dilute this stream on its way to the second column, so that this portion remains absorbed or gets absorbed at the entrance of the second column. Without the inline dilution, this portion would just flow through the second column and also elute all the starting material that has just been loaded onto the column and flush all of that into the outlet, so all the material would be lost. So the inline dilution is an essential part of this process. So now at the outlet of the first column, only the strongly adsorbing impurities are remaining. We are not interested in those, so we just clean this column and re-equilibrate it. And at the same time, we already start the gradient of on the second column to elute the weakly adsorbing impurities. Okay, so see, this happens simultaneously. And now this first column is re-equilibrated, and the process can start over just with the columns in the opposite order. Now, again, an overlapping portion is eluting from the second column. We want to keep it because it contains valuable product. So we interconnect the two columns. We elute this portion. We inline dilute it on its way to the first column now, so that it reabsorbs at the entrance of the first column. Then at the outlet of the second column, we have the pure product. We continue the linear gradient to elute the pure product. And then at the same time, we load new starting material onto the first column. And now again, this also happens simultaneously. It's shown here sequentially, but it's actually happening simultaneously. Product dilution and load of new starting material. Now at the outlet of the second column, we have the product overlapping with the strongly adsorbing impurities. We want to keep that fraction inside the process, so we elute this fraction with the columns in interconnected state, and we inline dilute this fraction on its way to the first column so that it gets reabsorbed at the entrance of the first column. Okay, here you go. So then at the outlet of the at the outlet of the second column, we just have the strongly adsorbing impurities left. So again, we clean the column and re-equilibrate it, and at the same time start the gradient on the first column to elute the weakly adsorbing impurities. And you've seen that short message one slide ahead. Here it was. At this stage, the cycle is complete and the next cycle can be started. So you see this is a cyclic process which can in principle go on forever. It has 2 product elutions per cycle, 1 from each column, and it has 2 feed injections per cycle, also 1 on each column. Okay, so this process, as mentioned, can in principle go on forever. We call this also a steady state. Steady state means product of constant concentration and purity is eluted from cycle to cycle. So you can't really tell if you're in the second cycle or in the 5th cycle. In practice, we see that it usually takes 1 cycle until the process has really reached this steady state, because we first have to internally recycle some side fractions, and then actually the profile, the internal profile of the steady state is established. But already usually in the first cycle we get purity of sufficient quality, because we are already cutting the peak and taking the center cut at the correct position. So also that material, although lower in concentration and yield, is already a good product that has product purity of sufficient quality. This process now can go on for many cycles, and in the end we can also have a shutdown where we actually recover this portion of the yield which is missing here. So it's not shown here, but through a shutdown cycle we can recover the missing product while we are actually not injecting any feed, any new starting material. In order to ensure that this process is operating robustly over also a longer period of time, we can use a UV-based feedback control that is essentially a collection of the product peak by threshold. So whenever the peak comes earlier or later, for example, through some temperature change, we can ensure that the peak collection and the recycling is done at the correct position. This is shown on this slide here. So M control is a UV-based dynamic process control that reduces the effect of temperature, also solvent quality, conductivity, pH, and also to some extent column variability, for example, through bed height differences. So the UV is constantly monitored by the software that's running the MCHCP process, and if the peak comes out at a different position, also the product collection will be started accordingly based on the UV values that the chromatogram reaches. Okay, let's move on to a case study for MCSGP. Here we are looking at a case study for an oligonucleotide. And the question is how to actually design the MCSGP process. Now we know how it works, but also the next point of interest is maybe how we actually get to MCSGP operating parameters, flow rates, gradients, and so on. So the process design procedure starts from a single column batch chromatography run that has some that we have carried out on the instrument that's running the MCHCP process, and that we have fractionated so that we know the distribution of the product and the impurities. Some of you know your processes by heart, so you know exactly where your product is pure enough and where you have impurities just by looking at the peak. So we need that knowledge. Once we have that, we can select the product range and recycling fractions by drag and drop and set the column size and the feed volume in a program which we call MCSGP Wizard, which helps us design the process. It's a guided procedure, and you here see the different tabs of this wizard. So this is actually the first page, that's the second page, and that's the third page. They're all shown here, one on top of the other. On the first page, you load the chromatogram with the fraction analysis that you see here, and then you select the product range, and by drag and drop, you define the region where you want to take out your product and the region of the chromatogram that you want to recycle in the front and in the back. And then in the second step, you define your washing and cleaning protocol. So here you essentially take the same protocol that you have used also for single column batch chromatography, right? So you just copy paste that essentially, and then you move on to the next step, which is some final settings. So here in this last page, you can enable the UV-based dynamic process control. You can set the number of cycles to run. You can set the mode of collection. Do you want to collect both columns together, the product from both columns together? Do you want to collect it separately? And also this last page will give you a prediction of the process performance. It's hard to see, but we will revisit this point later. Here you already at this stage get productivity predictions, buffer consumption prediction, before actually having carried out this run. So then once you've made your final settings, you push the save button, and then this wizard will generate methods that you then can load into the Condichrom system, which is carrying out the MCSGP run, and which you can then execute. Okay, a little bit a helicopter view, or zooming out, let's say. These are the chromatograms here that we have obtained from the single column run, and here you see the fraction analysis, and you see clearly it's a ternary purification, and for this oligo here, so we have weakly absorbing impurities in the front, strongly absorbing impurities coming off in the back, and the purity has a maximum, these are the dark blue lines here, dark blue crosses, the purity has a maximum somewhere in the middle, and it's lower in the front and in the back. The dashed line here is the purity specification of 90% purity. Okay, so this was anion exchange salt gradient. Now we're defining in the next step in the MCHCP wizard the lines for, or the sections of the chromatogram for internal recycling, product dilution, and internal recycling in the back. And we do that based on the product purity, right? So you see here in the center we have the product in specification. So obviously we want to choose that region as product pool, and the region that contains product but is below specification, we choose that for the internal recycling. And everything outside this region we do not consider. There's no product in there, so we don't need that, and we can discard everything that's outside this region here. Okay, then we enter the protocol for recovery and regeneration, and here we get do the final settings, number of cycles to be run, some pressure settings, and the fractionation. And then we generate them. So we generate the methods and we carry out the run. Here you see now the chromatograms from the run. You can see the conductivities and the UV signals. We expect to reach a steady state. So the UV signals at some point should all look the same. So we see alternating UV signals because we have one detector behind each column. So there will be peaks coming out in an alternating fashion from the system. And so in order to actually see if this process has reached a steady state, the software allows you to overlay the peaks. And here you see now an overlay of these 5 cycles that you've seen before, and it actually shows very nicely how the UV overlaps pretty well. The first cycle is a bit different, so here we have some disturbances. As I mentioned, it takes usually 1 cycle until the process is in steady state. However, very quickly the conductivities are matching up, and also when we actually compare the offline analytical data from the product purity, we see that we reach a steady state also very quickly. You see the offline analytical results here on the right-hand side, product concentration, purity, and yield, productivity, buffer consumption. Now also, I mentioned that the wizard can predict the process performance, and down here on the right-hand side, you can see the performance prediction by the wizard. You see the numbers are a bit larger here. This is because the wizard obviously assumes that we're getting 100% yield, which in reality, unfortunately, we're not getting. So a little bit of material is lost somewhere, which leads to a slight overestimation of the process performance by the wizard. You see the differences in yield is about 10%, and if you would account for that, multiplying the values here by 90.9, then you would also see that you would get a very similar values here in the productivity, which is also 10% off, and in the product concentration also. Okay, so when plotting the results of yield and purity, we see that we in fact have addressed the yield purity trade-off that we've seen from single column chromatography and moved to the upper right corner of the yield purity chart. Here we have 2 different single column runs. There's a benchmark run, and then there's an optimized batch run. This batch run was actually optimized in terms of productivity. So you see in terms of yield and purity, it's worse than the benchmark. In terms of purity, we'll see that it's better, but it's very far away from the MCSGP runs, which are in the upper right corner. So you see here MCSGP runs with different flow rates. They all end up in the upper right corner. So we can safely say, okay, we have improved the yield for from around 60% to 90 to 95% here using MCHCP as opposed to single column chromatography. Now, looking at the at the productivities, we see that we end up also in a similar range. So we are with MCSGP, we can improve significantly over the benchmark by a factor of 2. Now, if we take an optimized batch process, we can be higher in terms of productivity. But again, we sacrifice some material, some yield, as we've seen in the slide before. So if yield is really our key driver, then we are somewhere with the single column chromatograms, with a single column run somewhere in this range here, and then we are significantly better with MCSGP. Again, we are with MCSGP significantly better always, no matter how we optimize in terms of the yield. Okay, so summarizing, I hope I could show you how MCHCP can achieve high yield and purity in difficult center cut purifications, and that this yield leads to, first of all, improved productivity, which then leads to smaller columns required. It reduces solvent and buffer consumption. I think this is also obvious if I'm getting more material out, if I'm producing more product, but use the same amount of solvents and buffers, then the buffer consumption will be reduced. And also MCHCP, I think that's pretty clear, eliminates the need for rechromatography because we are getting a high yield. So there are no side fractions which are generated, and also no side fractions to be stored and analyzed, which then reduces the analytical burden. MCHCP can be applied to a wide range of products, including oligos, peptides, bispecifics, conjugated proteins, chart variants. We have application examples for all of these. So if you want to learn more about one of them, I'm happy to provide more data, and we also have publications on some of the topics here. Now, this first point here, I skipped that one when summarizing. That allows downscaling of the chemical synthesis, because I want to elaborate a bit more on this in the remaining minutes of this talk, because it actually has a very profound impact on the economics in oligo and peptide purification and manufacturing. So we have done an economic evaluation of the peptides manufacturing for the production purification of 10 kilograms of peptides. We have a white paper on this, so we can also provide this in case you're interested. The paper has been reviewed by some experts from the industry, so we are pretty confident that the assumptions that made in this economic evaluation are meaningful. I will not go through these assumptions, but here they're listed here, and you can review them later when you have a closer look at the presentation. I just want to mention a few key assumptions. So we compare in this paper MCHCP with single column processes which have 40, 50, 60, or 70% yield, right? As we've seen in the cases before, these are realistic values here, 60 to 70% yield in batch chromatography. Depending on the difficulty, it can be also worse, but these numbers are frequently seen in peptide and oligo purification. So we assume that with MCHCP we can obtain a higher yield, and we can assume that we also can use a 50% higher flow rate. This is because the MCSGP process has also not only interconnected phases, but also disconnected phases where the columns are operated on their own and where they have lower back pressure so that a higher flow rate can be used. Okay, so now this relates now to the synthesis costs that were mentioned before. So because MCHCP has a higher yield, you need fewer upstream synthesis batches to produce the same amount of material. If I'm wasting 50% yield in my downstream process, then of course I need to produce an extra batch to make up for that lost yield, and this extra synthesis batch will be very, very expensive. So so with lower yield, I need more synthesis batches to produce the same amount of product. And this, if we look at the cost of this, it's actually the dominating cost. We can calculate the payback period of MCHCP, and of course, compared to the different batch scenarios, the payback period is different, but in all cases, it's significantly less than 2 years, and actually less than 20 months also. So the elevated cost of procurement of the system capable of MCSGP is offset very quickly by the yield savings. Also, we have a significant saving in solvent consumption, which has an effect on solvent preparation and also footprint of solvent tanks. And here you can see the significant difference in solvent consumption of MCSGP versus single column chromatography. Okay, also column size is reduced by use of MCHCP. So instead of 70 liters of resin, we're talking here about 14 liters of resin in total, which allows to operate with much smaller columns and also allows to reduce the pump size on the skid. Okay, this shows now an overall summary of the cost. So here we have comparison of total costs of synthesis and the solvent cost, stationary phase cost, QA/QC, CapEx, plant operating costs, and you can actually see that the synthesis costs are really dominating. So it means that by reduction of increase of yield, reduction of chemical synthesis costs, we can save a lot of costs here. And you would expect that the major savings are on the actually purification side, but they are in fact on the upstream side. And here this difference, in best case, if we assume the best case of batch chromatography compared to MCSGP, the difference is still $600,000 US, whereas with worst case batch chromatography, it's 2.1 million for 10 kilogram of peptide. Okay, this looking at just the downstream processing costs. So this is without the synthesis. So the top part of these bars that you've seen before, we see that on the downstream side, we still save $200,000 US. This is mainly due to the savings in solvent costs and also stationary phase costs. The CapEx we see is a little bit larger here in MCHCP, which is due to the equipment costs. Okay, so with this, I hope I can show you the economic benefits of MCSGP also. It was very quick, but again, in case you would like to learn more, I would like to refer you to the white paper on this topic. And with this, I'd like to hand it over to Gerard for some final words.
Okay. Hey, thank you, Thomas. That was an excellent presentation. Now we all know that your passwords contain the acronym MCSGP. Talk a little bit about the background behind this technology, and that YMC Chromacon and YMC Process Technologies have been partners in scaling up from laboratory this technology from the bench scale on the left-hand side to full GMP scale systems, which are available today. The presentation was sponsored by YMC Corporation. You might know YMC is a leader in chromatography, producing both bench and prep scale HPLC and LPLC systems. packing resins, and columns since 1980. YMC has manufacturing and offices in over 10 countries. What you might not know is some recent expansion of YMC. YMC purchased both Chromicon and the former Leo Process Technologies, adding significantly to their 40-year history to now kind of lead the industry in the emerging area of multi-column or continuous chromatography. Okay, now I'd like to bring it back to Thomas for the question and answer. Thank you, Thomas, for your nice talk. I would like to ask you something about the flow rate. As far as I know, the flow rate used during the gradient in batch should be kept similar in MCSGP, but in which column? In the downstream column or in the upstream column?
Yeah, that's a very good question. So we always are conservative and say in the downstream column, the flow rate should be kept the same in single column chromatography and in MCHCP. And this is because in the downstream column, we will have the sum of the stream of the upstream column and the inline dilution. So we say both together should be equal or smaller than this flow rate in the single column chromatogram and the single column run to avoid any back pressure issues. Now you could argue that actually we are giving away some productivity here because we are only going through one column with that cumulative flow rate, which has half the bed height. But yeah, we're playing it safe here. We are conservative. So by ensuring this, we make sure that we never exceed the flow rate of the single column run. So we will never run into back pressure issues.
Thank you. Next question. Have you considered the lower cost of labor because there are fewer manual fractions to be collected and the packing and operation of the system requires less time?
Yes, so this is also a very good question. In the white paper, we have considered this by assigning a certain cost of sampling or cost per sample. We have not explicitly considered full-time employees or other cost of labor like insurance or whatever, because that very much depends on the location where the plant is actually set up or operating, and very much depends on let's say local on the local situation. So, but we've heard from users that let's say sampling or reduced sampling is a very important contributor. Especially, it appears that many users have let's say overwhelmed QC departments which are busy sampling. So it's a very important point. So we try to cover it as good as we can in the white paper.
Thank you. Now, here's a timely, relevant question. Can MCSGP improve production of RNA-based vaccines? That's also a very good question, and of course we've also been thinking about that recently. So we are currently initiating 2 studies with larger pharma companies where the aim is to purify RNA-based oligos using MCHCP. Well, we see, due to the wide applicability of MCHCP, we've used it for all kinds of molecules, even rare earth elements, so it's a general principle. Due to this general principle, we expect that MCSGP should also work for RNA-based oligos, and yeah, this could become very important when we think of capacity limitations with respect to RNA production that are currently in the industry. Okay, next question. Is there any pressure limitations for MCSGP?
So yeah, not really. So the equipment is designed in a certain way, and the pressure limitation, or let's say the pressure rating of the system, dictates the pressure that is then used in the MCHCP process. So HPLC equipment today can be well produced with ratings of 80 or 100 bar. And then the MCHCP can also operate within that pressure specification.
Okay, next, and I'm watching our time, so we'll take a few more questions. Thank you very much to everyone who's submitted questions. If we don't get to your question, we will respond in writing to you, and hopefully have a way to share the answer of that and the question to the entire audience in a follow-up email. Okay, what is the required similarity in column performance required for this system?
So we usually say the columns should be, as a rule of thumb, should be 10% similar. So you could run, let's say, a 10 centimeter column and a 9 centimeter column, or an 11 centimeter column within one system. We have not systematically investigated this, but that's just to our experience the kind of difference that we can afford. And I mentioned briefly that we have UV-based dynamic process control, and this UV-based dynamic process control will act if the peak from one column comes later or earlier than from the other, and will ensure that the collection is made at the correct point, so we can make up for differences in column bed height by using UV-based dynamic process control. And we can also design additional robustness into the process by selecting the product collection window a little bit narrower than we would need to do just based on the chromatogram. So essentially we have a little bit more safety with respect to a peak that's moving a little bit to the front or to the back.
Good question coming up, segueing into, I think your last response was, does the UV-based control require a special detector? No, so the UV-based control just works with the regular UV detector. So it uses a single wavelength, so either, let's say, UV 280 or 300 nanometers, and it just operates with that wavelength and reacts based on the single wavelength and the chromatogram of the UV trace at that wavelength. So answer is no special detector required. Okay, just 2 or 3 more and then we'll wrap up. During the MCSGP process, will I be concentrating my contaminants? Good question.
Yes, that's a good question. So we said we will reach a steady state at some point, and that also relates to the impurities. So when we internally recycle impure side fractions, we will get a higher concentration of impurities in the front and in the back inside the process. Now, however, also these impurity peaks, they will also grow outside of the recycling windows, as so to say. So the more impurities we accumulate, the larger the peak comes, but they also grow outside of the recycling windows, which means at some point we will reach the state where we elute the same amount of impurities in the waste fraction as we add through the feed material. So also to make a long story short, also the impurities will reach a steady state while they are actually during the process, they may reach a higher concentration than in the single column chromatogram or in the single column run. So eventually they will reach a steady state.
Okay, let's take this as our final question. Although there are many others, we can maybe issue an FAQ sheet because there are some very good questions that have come in. Is there a difference in how I should develop my separation if I want to use MCSGP?
Okay, so yeah, in some respect, yes, you should actually try to improve the productivity or to maximize more the productivity and the load. And you shouldn't bother so much about, shouldn't care so much about getting a high yield, because in MCHCP you will do the internal recycling of the impure side fractions. So if you have a process with, let's say, 50% yield, that's perfectly fine, because you can do the internal recycling of the impure side fractions and get to a high yield with MCHCP. So you can, let's say, put a focus on improving the load, put a focus on improving productivity, let's say by running higher flow rates or by running a steeper gradient. Right? So you may choose, let's say, different starting point for MCSGP than for let's say for single column chromatography, or let's say your starting gradient for MCHCP would be different from your final gradient that you are getting in single column chromatography.
Thank you very much, Thomas. Again, excellent presentation. Apologies for those who we did not get to your questions, but out of respect for everyone's time, I'd like to wrap up, and thank you for your participation today. Any of you have immediate questions or applications, you can reach out to us directly at sales@ymcpt.com. That's sales@ymcpt.com. We'll end our session now. Thank you and have a great day.
Bye-bye. Have a good day.